Good morning friends, welcome to my channel Medics Tutorial. This is the fourth video of the urinary system and today's topic is histology of the urinary system. So the urinary system consists of the kidneys, ureters and the urinary bladders and the urethra. The kidneys are vital in maintaining homeostasis of the body. They regulate blood pressure, blood composition and fluid volume of the body, produce and also produce urine and maintain acid-base balance, as we already discussed in the introduction videos of the urinary system. And in addition, the cells of the kidneys produce two important hormones. These are the renin and the erythropoietin. Renin regulates blood pressure to maintain proper filtration pressure for the kidneys, and erythropoietin promotes red blood cells formation in the red bone marrow. As you can see in this image, um, kidney kidney divides into the renal cortex. As you can see, this is the renal cortex, this is the renal medulla. In renal medulla there is a renal column, this is the renal column, and the renal pyramid, this is the renal pyramid. Now this is the corticomedullary junction in between the cortex and the medulla. As you can see, this is the renal papilla. This is the renal lobe, as it contains the pyramid, the renal column and the cortex. Fibrous capsule, which is the covering of the kidney. As you can see, this is the hilum, which is the concave shape. The renal artery enters through the hilum and renal vein exit through it, and also the ureter also exit through the hilum. This is the major calyx, this is the minor calyx, and this is the adrenal pelvis. Each kidney is bean-shaped with a concave shaped hilum, where the ureter and the urinary artery and veins enter. The ureter divides and subdivides into several major and minor calyxes, around which it located the renal sinus containing the adipose tissues. Attached to each minor calyx is the renal pyramid, a conical region of medulla delimited by an extension of the cortex. The cortex and hilum are covered with a fibrous capsule. Each kidney, each kidney contains 1 to 4 million nephrons, which is the structural and functional unit of the kidney. Each nephron originates in a cortex at the renal corpuscle, surrounding a small tuft of glomerular capillaries. Extending from the corpuscle is the long PCT, which leads to the short proximal straight tubules that enters the outer medulla. This tubule continues as the thin descending limb and thin ascending limb of nephron loop of Henle in the medulla. The loop of Henle ends with the thick ascending limb, a straight tubule that enters the cortex and ends at its thickened macula densa area, where it contacts the arterioles entering the glomerulus. Beyond the macula densa, this tubule is the distal convoluted tubule, the end of which is the short connective tubule. The connecting tubules from many nephrons merge into the cortical collecting tubules and collecting ducts that transports urine to the calyx. So, as we discussed the components of the kidney here, the kidney is the structural and the nephron is the structural and functional unit of the kidney. As the components of the nephron we'll discuss here. Nephron originates in cortex at the renal corpuscle. which are glomerular capillaries, tuft of the glomerular capillaries, which then leads to the proximal convoluted tubule, then the thin descending limb and the thin ascending limb of loop of Henle, then thick ascending limb and thick ascending limb, which leads to the distal convoluted tubule. After that, it ends in the collecting tubules. This collecting tubule merge to the other collecting tubules, which form collecting ducts. As you can see in this image, as we see earlier, this image, it is the magnifying image of this image here. So first we'll see it's a renal corpuscle. Renal corpuscle have glomerulus, which is the tuft of the glomerular capillaries. Also it has a glomerular capsule, which is lined by uh, visceral layer and a parietal layer. In between them it have a capsular space. As you can see, it is the parietal layer, and this is the visceral layer, and this is the capsular space. Also, this is the medulla, this is the cortex, and this is the medulla. As you can see, in medulla there is only collecting tubule meeting and forming a collecting duct. This is the proximal convoluted tubule as it arising from the glomerulus. After that, it originating the loop of Henle. As you can see, it is originating from proximal straight tubule. This is the loop of Henle, thin descending limb of loop of Henle. This is the thin ascending limb of loop of Henle, and this is the thick ascending limb of loop of Henle, which is giving rise to the macula densa, which is the thick macula densa cell, which is the and after that, this is the distal convoluted tubule, and this is the collecting tubule, which is entering to the collecting ducts. So we'll talk about the microvasculature of the renal cortex. As you can see in this image, this is the glomerulus, this is the afferent artery, this is the interlobular artery, and this is the peritubular capillaries, as you can see whole here. So the cortical vasculature is revealed in a section of the kidney with a renal artery injected with a carmine dye before fixation. Small interlobular arteries, as you can see, these are the small interlobular arteries branch from the arcuate artery and radiate out through the cortex, giving off the afferent arteriole, as you can see here, that bring blood to the glomerular capillaries. Each glomerulus contains a mass of capillary loops that drain into efferent arterioles, which draining into the efferent arterioles. This then branches has a large diffusive network of peritubular capillaries throughout the cortex, as you can see, peritubular network. Now we'll talk about the renal corpuscles. As you can see in this diagram, the afferent arterioles are enter into the glomerulus, and the efferent which forming the tuft of the capillaries, which is glomerulus, glomerulus, and which is entering out of the glomerulus by afferent arteriole. This is the vascular end where the afferent and efferent arteriole the enter and exit. This is the tubular pool where the proximal convoluted tubule is emerging out. So this is the Bowman's capsule or the glomerular capsule. So it is lined by a parietal layer of the glomerulus, as this is the parietal layer of the glomerulus, and the visceral layer of the endothelium, and both are lined. In between them there is a capsular space. As you can see here, this is the capsular space. So on the capillaries there is a pedicels are present, and also the podocytes podocytes cells podocytes cells are, as we show here on the visceral layer of the glomerular capsules. This is the endothelium of the glomerulus. As the afferent arteriole entering where and the distal tubule is meeting there, it forms a juxtamedullary apparatus, which is made up of the juxtamedullary cells and macula densa cells. So the renal corpuscles consist of a small mass of the capillaries called the glomerulus, housed within the bulbous cap, bulbous capsular glomerular capsule, which known as a Bowman's capsule, as you can see here. The visceral layer of the capsule is composed of a complex epithelial cell called a podocyte, which cover each capillary, forming slit-like spaces because interdigiting processes called pedicels. Blood enter and leaves glomerulus through the afferent and efferent arterioles respectively. So this image showing the microscopic graph of the Bowman's capsule or the glomerulus. So this is the parietal layer, Bowman's capsule. This is the capsular space, Bowman's space. This is the glomerulus. This is the macula densa. This is proximal convoluted tubule, and this is the distal convoluted tubule. The micrograph shows the major histological features of the renal corpuscles, as we discussed here now. The glomerulus of a capillary is surrounded by a corpuscular space covered by the simple squamous parietal layer of Bowman's capsule. Near the corpuscle is that nephron's macula densa, and section of proximal convoluted tubule and distal convoluted tubule, as shown in this diagram. Here we'll discuss the visceral layer of the glomerular capsules, which forming the filtration barriers. There is a podocyte cells on the capillaries endothelium, also it forming of filtration slits and pedicels. Filtrate is produced in a corpuscle when blood plasma is forced under pressure through the capillary fenestration across the filtration membrane or glomerular basement membrane surrounding the capillary, and through the filtration slit diaphragm located between the podocyte pedicle. As you can see in this diagram, this is the podocyte cell, this is the pedicle, and this is the glomerular capillary covered by podocytes with a pedicle. In this image you can see this is the filtration slits of visceral layer. This is the basement membrane of the capillaries, and this is the endothelium of the fenestrated capillaries, which form the filtration membranes. This is the lumen of the capillaries and the glomerular capillaries. This is the scanning electron microscopy, shows the distinctive appearance of podocyte and their pedicel processes that covers glomerular capillaries. As you can see, this is the this is the this is the glomerular capillaries, which is showing the podocyte on them and pedicel cells. Now we talk about the glomerular filtration barrier. The glomerular filtration barrier consists of three layered components: the fenestrated capillary endothelium, the glomerular basement membrane, and the filtration slit diaphragm between the pedicels. The major components of the filter is formed by a fusion of the basal lamina of the podocyte and the capillary endothelial cell. This image this image shows cell bodies of two podocytes. As you can see here, this is the cell bodies of the two podocytes, and the series of pedicels on the capillaries. This is the capillary, and this is the basement membrane separated by the filtration slit diaphragm. Around the capillaries and podocyte in the capsular space. This is the capsular space into which the filter enters. As you can see, this is the two podocyte bodies. This is the capillaries, and this is the pedicels present on that. This is the capsular space, and this is the basement membrane. At higher magnification, both the fenestration in endothelium of capillaries and the filtration slits separating the pedicels are clearly seen on two slides of thick fused basement membrane. This slit diaphragm bridged the slit between the pedicels. As you can see here, this is the filtration slits. These are the pedicels. This is the basement membrane. This is the endothelium. This is the capillaries. This is the These are the fenestrations of the endothelium of the capillaries. These are the slit diaphragm, and this is the filtration slits. This diagram shows three parts of the glomerular filter and their major functions. As you can see in this image, this is the fenestrated capillary endothelium. This one is the glomerular basement membrane, which blocks the large proteins, and this is the pedicels which forming the filtration slits diaphragm between pedicels, which regulates passage of many small proteins. Mesangium. Messenger cells. Uh, in messenger cells are the cells present in renal corpuscles, are located between capillaries and cover those capillary surface which not covered by podocyte processes. As you can see in this diagram, this is the podocyte here, and this is the capillary, and here is the capillary present. Now here is a capillary, and here is capillary, and here there is no podocyte available. So instead of podocyte, here messenger cells are present. In this image shows one messenger cells. This is the messenger cell, and surrounding messenger matrix. As you can see here, this is the messenger matrix. This is the messenger matrix. This is the messenger matrix. This matrix appears similar to and in many places continuous with the basement membrane. As you can see here, it is continuous with the basement membrane and support capillaries where podocytes are lacking. Messenger cells extend contractile processes. As you can see here, which extend the contractile processes along capillaries that help regulate blood flow in glomerulus, that regulate blood flow in a mesangial cell, regulate blood flow in the glomerulus. Some mesangial processes appear to pass between the endothelial cells into the capillary lumen. Asterisks, that's known as asterisks, where they may help remove or endocytose adherent protein aggregates. Also, the second function is it helps to remove or endocytose adherent protein aggregate. The capillary at the left contains an erythrocytes and lymphocyte, as you can see here. Capillary contain erythrocytes and lymphocytes here. Podocyte cells, as you can see, this is the podocytes, this is the podocyte, and their pedicels. These are the pedicels open to the urinary space. This is the urinary space. It is open into the urinary spaces now, and associated with the capillary surfaces not covered by mesangial cells. Slide you'll see In this slide you'll see the histological features and major functions of regions within the renal tubules, such as the PCT, loop of Henle, collecting system, etc. So first we'll talk about the PCT, proximal convoluted tubule. It has simple cuboidal epithelium cells are well stained with numerous mitochondria. It have prominent basal folds and lateral interdigitations. Also have a long microvilli, lumen often occluded. So we can see the lumen is very small or uh, not able to see. Mm, its location is mostly present in the cortex and function. It works by reabsorption of all organic nutrients, all proteins, most water, and electrolytes, secretion of organic anions and cations, hydrogen and NH4. Now we'll talk about the loop of Henle. First we'll talk about the thin limb. It has simple squamous epithelium as the area as it's a thin limb, no, so there is a less area to accommodate the epithelium. That's why it get adapted in a simple squamous epithelium in thin sheet. Also it have few mitochondria. It's present in medulla and works as a passive reabsorption of sodium and chlorine. Thick ascending limb. It have simple cuboidal epithelium, no microvilli, but many mitochondria. It have many mitochondria. It present in medulla and medullary rays. It performs function of active reabsorption of various electrons. Distal convoluted tubules. It has simple cuboidal epithelium, cells smaller than PCT, short microvilli, and have basolateral folds, more empty lumen. As compared to the PCT and DCT have a greater lumen. It also present in cortex and it works as a reabsorption of the electrolytes. Now we'll talk about the collecting system. It have principal cells, which is most abundant, cuboidal to columnar, pale staining, distinct cell membrane. It present in medullary rays and medulla. It regulates reabsorption of water and electrolyte, regulates secretion of the potassium. P for principal, P for potassium, P for principal, P for potassium. So you can remember that. Now intercalated cells, few and scattered, slightly darker staining, presents in medullaries. It works by reabsorption of potassium. Those who are taking the potassium diet and helps maintaining acid-base balance. So that's it for. Now I'll talk about the renal cortex and mainly the proximal and distal cannulated tubule. In this micrograph, we'll see the Bowman's capsule and the associated structure like PCT, DCT. This micrograph shows the continuity at a renal corpuscles tubular pole. This is the tubular pole from where the tubules are arising between the between the simple cuboidal epithelium of the proximal cannulated tubule and the simple squamous epithelium of the corpuscular capsule's parietal layer. The urinary space, as you can see here, this is the urinary space or the corpuscular space or Bowman space between the parietal layer of and the glomerulus drain to the lumen of the proximal cannulated tubule. It is draining to the proximal cannulated tubule, as you can see here, like that. The lumen of the proximal tubules appears filled because of the long microvilli of brush borders and aggregates of small plasma proteins bound to this structure. As you can see, this is the proximal cannulated tubule. This is the proximal cannulated tubule, and this it's appear like it's appear filled because of it have long microvilli of brush borders and aggregate small plasma proteins. So it's appear like a field. That's why it's a, so you can identify it from the DCT. Now we'll talk about DCT. The lumen of the distal convoluted tubule, as you can see here, it appears empty. As you can see, it appears empty because lagging of the brush borders and a protein. So now you can distinguish between a PCT and DCT on basis of their lumen. If it's filled, then it's a proximal connective. If it's empty, then it's a distal connective. Here, the abundant peritubular capillaries and draining into the venules. As you can see here, these are the venules, and these are draining by the peritubular capillaries, surrounding by the proximal and distal convoluted tubules. As you can see, they are surrounded by proximal. These are the proximal, and this is the distal. As you can see, distal convoluted tubule is empty lumen, and the proximal convoluted tubule is filled, filled, filled lumen. This is the peritubular capillary draining to the venules. Sorry. Now we'll talk about the leaf of Hindley. As you can see in this image, it is a diagram of nephron, shows levels of section in the photos. As this, we take first section here, cross section of this part, and magnifies it as like here. So we see it. We take a section of cortical tissue shown one renal corpuscles. This is the renal corpuscle. The wide eosinophilic proximal convoluted tubule. This is the wide As you can see, this is the wide eosinophilic proximal convoluted tubule with the smaller and less well-stained distal convoluted tubules. This is the distal convoluted tubule, which are the smaller, less well-stained distal convoluted tubules, as you can see in this image. Now, this diagram shows major structural difference between the cuboidal cells of proximal and distal tubules. Cells of both tubules have basal membrane invaginations associated with the mitochondria. As you can see, this is the proximal convoluted tubule and this is the distal convoluted tubule. It have nucleus, mitochondria and basement membrane. But the only difference is proximal convoluted tubule have tall microvilli which make them which no, which make them filled and it gets filled appearance, and distal community will have short sparse microvilli. They are short, that's why it appears empty. Now after that we'll take one more section from here and we magnify it here and we'll see a cross section through medullary renal pyramid. This is the medullary renal pyramid section, shows the simple squamous epithelium of the thin descending and ascending limb of loop of Henle and its thick ascending limb, as well as pale columnar cells of the collecting ducts. As you can see here, these are the collecting ducts. This is the thick ascending limb of loop of Henle. This is the thin descending limb of loop of Henle. Note also the homogeneous interstitium with capillaries smaller than thin limbs. We'll see the Now we'll see the ultrastructure of the proximal convoluted tubule. The TEM, as we can see, the transmission electron microscopy of the proximal convoluted tubules reveals important features of the cuboidal cells of the proximal convoluted epithelium, which is a large, dense, which have large, dense apical microvilli, as we can see here, which is the large, dense apical microvilli with abundant endocytic pits and vesicles, as you can see here in the apical region near the lysosome. As you can see, vesicles are near the lysosome. These are the lysosomes. Small proteins brought into the cells, known specifically by pinocytosis. Do remember, small proteins brought into the cells by pinocytosis are degraded in lysosomes and the amino acids released basally. In apical ends of adjacent cells are filled with the zonula occludens, but the basolateral sides are characterized by long invaginating folds of membrane along which may, many long mitochondria are situated. As you can see, these are the mitochondria. Water and small molecules released from the PCT are taken off immediately by the adjacent peritubular capillaries. As you can see, these are the peritubular capillaries. Between the Between the basement membrane and the tubule and the capillaries shows an extension of the fibroblast. As you can see, this is the extension of the fibroblast in between the basement membrane and the capillaries. Renal medulla, in which we'll talk about the nephron loops and the collecting ducts. It is a micrograph of medullary renal pyramid cut transversely, shows closely packed cross sections of the many nephron loops, such as thin descending and ascending limbs, as you can see here. Thin descending and the ascending limbs, and thick ascending limbs. These are the thick ascending limbs, which are the intermingled with parallel vasa recta capillaries containing blood. These are the vasa recta capillaries and the collecting ducts. As you can see, these are the collecting ducts. These are the collecting ducts. All these structures are embedded in the interstitium. As you can see, it is the interstitium, and all the structures are embedded in the interstitium. Interstitium, which contains sparse microfilament-like cells, and a matrix which is very rich in the hydrophilic hyaluronate, which is very rich in the hydrophilic hyaluronate. The specialized nature of the interstitial tissue is helped to maintain the osmolarity gradient established by different salts and water transport across the walls of nephron loop, which is required to concentrate urine and conserve body water. The TM reveals the slightly fibrous nature of the interstitium. This is the interstitium, which is in fibrous nature. This is the interstitium that shows the simple squamous epithelium of thin limbs. As we see, this is the thin limb which have the simple squamous epithelium, as I'm showing here, which is slightly thicker than that of the nearby vasa recta capillaries. These are the vasa recta capillaries. So the squamous epithelium of the thin limb is thicker than that of the vasa recta capillaries. Juxtaglomerular apparatus. The juxtaglomerular apparatus forms at the point of contact between nephron's distal tubule and the vascular poles of its glomerulus, as this is the vascular pole of the glomerulus, as the afferent and efferent arteries entering to the glomerulus, and this is the distal tubule of the nephron, which is contacting to the vascular pole, and this is the juxtaglomerular apparatus in between the uh, contact of the vascular pole and the distal convoluted tubule. So at the point, cells of distal tubules become columnar, as a thickened region called the macula densa cell. These are the macula densa cell. These are the macula densa cell which become columnar as thickened region called macula densa, and the smooth muscle cells of afferent arterioles tunica media are converted from contractile to secretory morphology as juxtaglomerular cells, JG cells, as you can see here. Also present lytic cells, which, as you can see here, which is lytic cells, which are extraglomerular mesangial cells adjacent to the macula densa. As you can see, it is adjacent to the macula densa, and the afferent arteriole and efferent arteriole. As you can see, adjacent to the afferent and the efferent arteriole. In this specimen, the lumen of the proximal tubule appears filled. As you can see, this is the lumen of the proximal tubule which appears filled. As you can see here, it appeared filled, and the urinary space is somewhat swollen. This is the urinary space which looks like it's swollen. So mainly the juxtaglomerular apparatus is formed by the macula densa cell of the distal convoluted tubules and JG cell of the afferent arterioles, and the lytic cells are also present there, which forms the extraglomerular mesangial cells, which are adjacent to the macula densa, then the afferent and efferent arterioles. The collecting duct, pill staining columnar principal cells in which ADH regulated aquaporins of the cell membrane allows more water reabsorption and are clearly seen in this transversely section collecting ducts. These are the collecting ducts. These are the collecting ducts surrounded by interstitium with the vasa recta. As you can see, this is the vasa recta. Renal papilla, collecting duct, and the minor calyx. It is as you can see this image, it is a sagittal section of the renal papilla, shows numerous collecting ducts, which is also called the ducts of the Bellini, covering at the end of the renal papilla, where they empty into the minor calyx. As you can see, this is the renal papilla, and this is the minor calyx, and this is the collecting duct. The mucosa of the calyx contains dense connective tissues stained blue here, and adipose tissue. As you can see, these are the adipose tissue, and this is which is showing the blue, no, that's a dense connective tissue. The duct are embedded in interstitious tissue, which also contains thin limbs of the loop of Henle. An enlarged area, as we show, this is the enlarged area which is magnified here, shows the columnar epithelium of the collecting duct. This shows the columnar epithelium of the collecting duct. The interstitium, as you can see, this is the interstitium, and the thin limb. This is thin limb of the nephron loop, and the protective urothelium that lines the minor calyx. This is the protective urothelium, layer which pro- which protecting the urothelium. Ureters. In this diagram we can see the cross section of the ureters. So in this section we will see the layers of the ureters. So innermost layer is the mucosa, then the muscularis and the adventitia. Again the mucosa have two layers, transitional epithelium and the lamina propria. So the innermost is the transitional epithelium, then lamina propria, then the muscularis and the adventitia. So diagram of ureter in cross section shows a characteristic pattern of longitudinal folded mucosa surrounded by thick muscularis that moves urine by regular waves of peristalsis. The lamina propria is lined by unique stratified epithelium called the transitional epithelium or the urothelium that is resistant to the potentially deleterious effect of contact with hypertonic urine. As histologically, the muscularis, muscularis layer is much thicker than the mucosa and the adventitia. As you can see, this is the mucosa, this is the muscularis, and this is the adventitia. As you can see, the muscularis is the much thicker than both mucosa and the adventitia. Bladder wall and the urothelium. In the neck of the bladder, near the urethra, the walls shows four layers. As the first one is the mucosa, second one is the submucosa, third one is the smooth muscles, and the fourth one is the adventitia. As you can see, first one is the mucosa, second one is the submucosa, third one is the smooth muscle, and the fourth one is the adventitia. As you can see, adventitia, smooth muscles, then the submucosa, and this is the mucosa. Got it? Four layers. Then then we see some points regarding each layers. The mucosa have the urothelium. As you can see, it have a urothelium here, and the lamina propria layer. Mucosa have urothelium and the lamina propria. Got it? Then the submucosa is the very thin as compared to other layers. As you can see here, this is the submucosal layer, which is the thin. Then there are three layers of the mm, smooth muscle. First one is the inner layer, second one is the middle layer, and third one is the outer layer. Got it? And then lastly, there is the adventitia. It is from outside the inside. First one is the adventitia, smooth muscles, sub submucosa, then the mucosa. Again, adventitia, then outer layer, middle layer of smooth muscles, and the inner layer of smooth muscles. Then submucosa and lamina propria of the mucosa and the urothelium of the mucosa. Got it? When the bladder is empty, in this image you will see what happened to the urothelium when bladder is empty. The mucosa is highly folded and urothelium has bulbous umbrellar cells. As you can see, the mucosa is folded here as you see, and the urothelium is bulbous and umbrella shaped as you can see here. So remember, when bladder is empty, the mucosa is highly folded and the urothelium has bulbous bulbous umbrella shapes. And when bladder is full, the mucosa is pulled smooth as you can see here. It's pulled smooth and the urothelium is thinner and the umbrella cells are flattened as you can see here. The urothelium is the thinner and the umbrella cells get flatter. Urethra. Now we'll talk about the urethra. It is a fibromuscular tube that carries urine from the bladder to the exterior of the body. The male urethra is longer and consists of three segments as follows. First, we'll talk about the prostatic urethra. It is 3 to 4 centimeter long, extends through the prostate gland, and is lined by urothelium. Second is the membranous urethra. It is a short segment, passes through an external sphincter of striated muscle and is lined by striated columnar and pseudostratified columnar epithelium. A spongy urethra. It is about 15 centimeter in length, is enclosed with erectile tissue. It is enclosed with the erectile tissue. Do remember that's a spongy urethra of the penis and is lined by stratified columnar and pseudostratified columnar epithelium with stratified squamous epithelium distally. In women, the urethra is exclusively a urinary organ. In female, urethra is 3 to 5 centimeter long tube lined initially with transitional epithelium, which then transition to non-keratinized stratified squamous epithelium, continuous with that of the skin and the labia minora. The middle part of the urethra in both sexes is surrounded by external striated muscular sphincter, as we seen here also. Membranous urethra is the middle part of the male urethra, which is pass which is passes through external sphincter of the striated muscle, and in in female urethra the middle part is also passes through the external striated muscle sphincter. This is the image of transverse section of urethra. Shows the mucosa has large longitudinal folds as you can see here around the lumen. This is the lumen. And this is the higher magnification of the enclosed area as we saw here. Shows the unusual striated columnar nature of the urethral epithelium as you can see here. This thick epithelial lining varies between striated columnar in some areas and pseudostratified columnar elsewhere, but it becomes striated squamous at distal end of the urethra. Now we'll talk about the fluid transport in the urinary system. it means the pathway of the urine. So first blood entered into the glomerulus. After the filtration, the fluid enters into the capsular space. It's known as the filtrate. So after filtrate enters to the capsular space, which is mentioned here, it enters to the proximal convoluted tubule. This is the proximal convoluted tubule. After that, it enters to the descending limb of nephron. This is the descending limb. After that, it enters to the ascending limb. Then it enters to the distal convoluted tubule. After that, it goes to the collecting tubule, as you can see here. After joining collecting tubules, it forms a collecting duct, and filtrate enters to the collecting duct, as you can see here. And after that, it enters to the papillary ducts. After entering into papillary duct, filtrate will call the urine. So after papillary duct, it goes to the minor calyx, as you can see here. Then it goes to the major calyx, then renal pelvis. After that, the ureter, then urinary bladder. After that, urethra, and from urethra it gets passed outside the body. Here we end with the histology of the urinary system, and the next video will be on physiology of the urinary system. Thanks for watching. Like, share and subscribe.